show the Jigyasa website and talk about it.
Just
अरे
you
नहीं
know
नहीं
it.
नहीं
No?
नहीं नहीं
अरे अरे
नहीं
बोल
ma'am,
दो
नहीं नहीं
बोल.
नहीं नहीं
Come on.
I'm I'm not prepared for that, man.
There's nothing to prepare. Just see what the activity is and what we are doing in this session. That's it. Somdatta.
I anyway do, and I did last time also. I will do the next time also. So if you're here, I would recommend one of you take it up.
Sridhar, go ahead.
Awesome.
Ma'am, next time, ma'am, next time.
No, next time. Go ahead.
Good morning, everyone. Welcome to the second talk in the series of Futuristic Fridays, AI in Biology and Health, which is being coordinated by CSIR HRDG in Delhi and CCMB in Hyderabad. And in this series of talks, we will be meeting a series of experts talking about AI in biology and health. And today in this series, we have Dr. Lipi Thukral from CSIR IGIB, which is also based out of New Delhi. And while you will get to know about the speaker and the series and what she wants to talk about in a little while from me, I will request Dr. Sridhar, who has joined us from CSIR HRDG today, to tell you about the CSIR's Jigyasa program. A little bit to you first, and the talk series, the Futuristic Fridays talk series, is a part of the Jigyasa program. Sridhar, may I request you to take over the screen?
Yeah, hi, good morning. Good morning, Somdatta. Good morning, Dr. Lipi, and good morning, everyone. Thank you for joining this series. To talk about Jigyasa, it got started in 2017. It's a Student Scientist Connect program, which is a scientific social responsibility of CSAR. Uh, Basically, we try to connect with school students through various programs like scientists visiting labs or students visiting our CSI labs. And we do a lot of experiments. We do a lot of activities like, hands-on activities. And one of the, key initiative is the Futuristic Friday, where we try to, bring in scientists to talk about their research work, their, experience as their journey as a scientist from their college days till their scientific work. The first futuristic Friday talk was held on 5th, and today is the second one. So I believe that this talk will be a very informative as well as will motivate many students to take up science as their career. So we expect a lot from Dr. Lipi, and we expect that you may connect with students and brief about how AI can be used as a future career, and especially in biological science. Thank you. Thank you.
Thank you so much, Sridhar. And I must also add that students who have joined us, you should definitely be registering for this Futuristic Fridays series. You're seeing the screen and the web link there. If you go there, there's a button called as Participate Now. And when you do that, for every talk, you'll also have a quiz. And by participating in those 4 quizzes, because we have 4 talks and hence 4 quizzes, some of you will also be selected for a virtual week-long program where we want you to be participating in a week-long project. So do participate in this. And in addition to this Futuristic Friday series, Jigyasa does a lot of other programs, which includes the physical programs like where scientists go to schools and colleges, as well as students come to our labs and see what real life scientists do, what kinds of questions they address, why they work on these things. And in addition to that, we also have a lot of online digital content, which you are seeing on the screen right now. And they range from games to videos to podcasts to simulations. And the hope is that while it might be difficult for every student to come to a lab and see what happens in a lab, but these content should help our classrooms talk about science in a more fun, engaging, and real life driven way. With that, let me come back to the talk for today. And to my great pleasure, I will be introducing Dr. Lipi Thukral, who comes from CSIR IGIB. Lipi works on understanding structures of molecules inside cells. Cells are filled with all kinds of molecules. Proteins and lipids are some of the kinds of molecules that she looks at. And these are huge, present in huge numbers, and each have different unique structures. And understanding them should already give you an idea of how much of data that she is working with. And that's where really her skills with computational biology comes in. And she tries to make sense of the structures of these molecules and what happens to them when we are suffering through some diseases. So with that, I'll hand over the mic to Lippy, and we will like Lippy to first tell us about a little bit about her career trajectory so that our young people see how different kinds of backgrounds and motivations are working in this interface of AI and biology and health. And then we will deep dive into what she is doing in her lab. Lippy, over to you.
Thank you so much, Somdatta, and thank you so much, Sridhar, for organizing this Futuristic Friday. And I'm really grateful for CSIR Jigyasa program to connect with so many initiatives with students. So thanks a lot for doing this. And yeah, now to the students who are hearing me, hello everyone. Lippy Thukral. I'm a computational biologist. What does that mean? That's what my job is to it in this 1 hour and tell you that what we do in the lab. So although the series is Futuristic Friday, my idea is also to take you back a little bit when we were not doing so much of computational biology, when the biology was still pen and paper. Can you go back in your minds 20 years back? But maybe at that time you were not born, right? Or maybe, I can just provide you with the glimpse. So you can walk through my eyes, you can walk through my memories. Just 20 years back, when biology was very going through a transition phase through modern biology tools, there were a lot of tools that were coming up. People had just started doing genomics. The first human genome draft was happening. So people were just doing back and forth Excel sheets. So imagine now there's a little bit of data coming. The databases were just there. And at that time, I was a young student who was doing an undergrad in biotechnology. And I felt completely out of the place. I thought that this career is not for me. I'm doing biotechnology, but I don't know if I can stand up and do so many experiments. So that's where I tried to study about these data sets that were coming up. NCBI, which was a pioneer in making those data sets, was doing all sort of compilation. It was just trying to put up a word called database for every kind of biological data. So we were exploring that. There were algorithms that were coming up like BLAST. People were trying to understand if there is a protein sequence, can we align them? What kind of alignment tools are there? This was just 20 years back. And I was this undergrad student who was just wanting to understand how can I use another set of technique to study biology, because experimental biology was just not hitting for me. So I went for training in different places, in different labs. That's what I think as a young student, if you have the chance and you already can see this, that you're getting explored through various channels through Jigyasa, what is science about? So science is not about one field, right? So that's where it all started for me. So I went through different internship programs and I realized that there is a extremely specific field which had developed by then called bioinformatics. And that made me shift from biotechnology to do masters in bioinformatics from Banasthali Vidyapeeth in Rajasthan. And I just knew that I am completely in my zone where I'm passionate about doing things on computers, seeing the data on computers, writing algorithms. So I used to take evening classes of computer programming when I was in undergrad. So it just came together. And a lot of practicals we would do, a lot of programming we would do. That's how I started my journey in computational biology. And then I did a short training in IIT Delhi with the Professor Jairam. That's where it just like to understand how is the research done. That was my first stint in research. And from there, I got accepted in a PhD program at University of Heidelberg, where with Professor Jeremy Smith, I entered more specifically in a niche area of molecular dynamics simulations, and they're trying to understand how proteins fold. Now, this has been overall my educational journey. And then, of course, after PhD, I have done my postdoc and then came back to India to sort of open this field here and try to still do interdisciplinary research. Now, what is interdisciplinary research? Interdisciplinary research means that you can still come from very, very orthodox departments like physics, chemistry, and your research can be from these departments, but then you apply your knowledge and you use other interfaces like chemistry, computers to still apply doing solving biological problems. So this is where my lab is focused at IGIB, and I'm going to show you that in through my slides now. So just a moment. So it's been 10 years now that I am at IGIB, and what we've been doing is that we've been looking at structures. So let's see if I can just get a confirmation from folks here. Can you see my slides in slideshow mode, please? Can you?
Yes.
Okay, brilliant. Okay, so yeah, so I hope with that background, I'll be able to today give you an idea about how much advancements since 20 years we have made in structural biology, and how AI tools, and one of the AI tools, at least in structural biology, called AlphaFold 2, has been a paradigm shift in doing how we do things and how we analyze things and how we generate data in structural biology. So let's get started. I am here in IGIB Mathura Road campus, which is a beautiful campus, and we are sitting here right now when I'm giving the talk on second floor. This is my lab, and day to day we do come here and we sit on our computers to do research. Today, as I said, is a special day because I'm connecting with school students. We are trying to give you a glimpse that every day what we do. Apart from me, we are also a very thriving computational biology unit at IGIB. So we are not just studying structures as my lab does, but there are also people who are doing healthcare monitoring. There are people who are doing medical informatics, genome sequencing, of course. So with this slide, I wanted to give you an idea that computational biology more uniquely you'll understand from here, from my expertise, but there are also other kinds of fields which are available, which give you very different kind of information. That is number one. For example, if a patient comes in and tells, yeah, these are my measurements, these are mostly saved in text-based data, so even the compute need is very different. So you get phenotypic level data, okay, and you get images, you give images, that is also phenotypic level data. And you have image-based, so the compute need slightly increases. The compute need, what do I mean by compute need? You need to process this data, right? So a small laptop is not sufficient. And then as we go to genomics, the compute need even increases in order of magnitude. So you need more and more computations, and also the data that is generating is a large. And where I am sitting, I utilize supercomputers. So you can consider supercomputers to be stack of computers on top of each other. And you can use now thousands of cores to run the simulations or to analyze the structures. So this is where the compute need is very, very high, because the data is not text-based, the data is not image-based, but it's multi-dimensional. It has also time series data. So it's very interesting to write algorithms and to also analyze this data. It becomes very, very challenging also sometimes. So let's give you some of the keywords. As you can now understand that even using computational biology, there are different techniques. So with that, if you are studying an X disease, let's say any kind of disease, cancer disease, now in biology, there are different kind of readouts. What I mean by readouts is that X technique is giving you a different readout and another kit is giving you another readout. So there is a lot of differences in kind of data which is emerging. It's not like the same. It's not like Paytm where, you know, there's just one kind of giving money kind of a data. You have every technique, and the techniques are even evolving every day as we speak. So there is a lot of heterogeneity. And also what is happening is that the scale of this data is different. The resolution of this data is different. So what you need to take away from my first part of the introduction is that data is not a bunch of points anymore. So there have been huge technological advancements which have made data very complex. So therefore, we need folks from different backgrounds to take care of data analysis, getting data started. My part of the world, we study protein structures, which are on the left hand side. Protein structures are something that directly link atoms to function. So for example, if you're moving your body, there is a protein called myosin, which is present in the muscle responsible for the movement. As I speak, as I inhale, as I have infection, there are proteins which are responsible for these activities. Okay, so we try to, using studying protein structure, link this to function. And in recent months or recent year or so, there has been remarkable increase in structural biology. And I would like to now assign this increase to three significant areas. One is electron microscopy. And I'm going to come to all of these in a bit. The second is the AlphaFold 2, which is a program developed by Google DeepMind, a company in London. So it's not an academic effort. It was one of its kind where company has developed a program which superseded everyone's expectations. And the third is high performance computing. I just briefly mentioned you that the program that we are using are large supercomputers. So, you know, the computers are now, and you would know this, that you can see the movies on your mobile. So what mobiles have, the RAM, their speed, their ability to process so many apps together was not there 10 years back. Similarly, what we have in terms of our compute cluster is very, very different than what we were having before. So all this combined, we are able to study biological length scale at very interesting way. And I want to just show you a picture to give you an idea about size. Okay, what I mean by size. So let's start with electron microscopy. The electrons, so it's here, but I'm going to come back in a while. Humans can be studied in meter time scale. Okay, so let's say we'll have some crude measurements to study humans. Then mouse comes in 10 centimeter. We have a couple of other microorganisms that fall in millimeter. But now these are all in naked eye. So you can study them. Okay, there are fields which study them through just naked eye measurements. But let's go into modern biology, where a lot of cells are studied with microscopy. Okay, they can handle micrometer, 100 micrometer. Red blood cells are, for example, 6 to 8 micrometer. And mitochondria, for example, is 0.5 to 3 micrometer. This is very, very interesting because as you go even smaller, you are at nanometer. You know what is the tiniest nanometer you have? That is an atom. Beyond atom, you don't have anything. So it's like you cannot go further. You also cannot see atoms right now in electron microscopy. But what electron microscopy can handle is that it can show us viruses. You know how crazy viruses are? They are completely doing a lot of messy biology in our bodies. It introduces a lot of infections. And as you all know, during COVID, SARS-CoV-2 was the predominant factor, and this was routinely studied using electron microscopy, which can handle easily a virus particle. Then there are also things which are inside our cells, like some vesicles, some proteins of certain dimensions. As I said, beyond certain dimensions, electron microscopy is incapable, like small molecules, small proteins. So this is still where completely it's not there. We are getting very, very close, but yeah, it's still not there. However, there are other techniques which we can also do in this length scale. So this is mostly another extension of the previous slide, where I'm trying to show you that how other techniques, particularly in structural biology, like to study single molecules, proteins, we do that. Okay, so the length scale here is here. I'm introducing a component called time, because time is very interesting for me. Every cell, proteins move. So There are techniques like electrophysiology, FRET, NMR, AFM that study proteins at the length scale I was talking about, which means smaller proteins to large proteins. There is, of course, EM and X-ray, which are studying from small protein to larger proteins. But look how interesting is it. We can also study time. You know why? Because proteins move. We move. Inside us, everything moves. So we need techniques that can make structures, atoms, all these things move to allow us to study them. So one of them is then molecular dynamic simulations that I'll come to, high performance computing that I'll come to. And what are the things? I'll give you 4 examples of my own work to study this. But before that, I want to tell you a just very important thing, that if you are interested in anything what I say and later you want to go back, this is the page you should go back to. So write on Google PDB all the structures that are studied using these techniques, especially the X-ray crystallography and electron microscopy, are deposited in a database called Protein Data Bank. This is probably, to my knowledge, the first structured database in general in biology. So the PDB has been very, very progressive in putting together whoever does any kind of experimental technique and produces 3D coordinates, including NMR, X-ray, and now EM, can deposit the structures here. So if you're interested in looking at the structure and you're very curious, then you should go back to PDB and you can now type in any protein. Let's say I have written tubulin here. You can write hemoglobin, right? Hemoglobin is a protein. So if you know any protein name, you can simply type in here, please. And you can get pages like this. So I'm seeing an alpha beta tubulin page. There is a 4-letter code here. And you can see the protein structure here. You can download the file. This will be in .pdb format. And there are tools like Chimera. You can write down Chimera. You can write down VMD that allows you to see these tools. Okay. And I'm someone who's fascinated by structures. So I see structures the whole day. But if you're just interested in seeing more information, everything, there are so many tabs, please go and explore. But once you download file, I'll just give you one last information, because it's not a demo. I will still like to cover other things. This is a PDB format. Okay, so you get columns like which has atom number, all these coordinates that give you the exact location. So this is the structure PDB format. Okay, and don't be too daunted by it. It's just that there is a very specific format which allows us to process this. So anybody of you who has taken computer science along with biology, or in general has mathematics and computer science as well, you can look at these formats and you can ask different questions. That can I just fetch ligands from it? After my talk, you'll have more clarity. But this is the file which you can play around with. Okay, so let's move on. Yeah, this is the structure, how it looks. So if you can now manage to see that using VMD, you can also look that this is beta tubulin, this is alpha tubulin. Using the PDB format, you will get to know how many amino acids are there, what is the resolution, and so on and so forth. Okay. Now coming to the part which I'm very excited, and we have this AI series, is about a tool called AlphaFold 2. And again, before coming to AlphaFold 2, I want to just explain to you that prior to AlphaFold 2, what we were doing very fundamental prediction of proteins which maybe did not have a good confidence score. So what happens is that in homology modeling, you try to model a protein which is not available using any experimental technique. Right, so we were doing template-based modeling, fragment-based modeling, ab initio modeling, and these are all primarily based on how much available sequences were there prior to it. Now, AlphaFold 1 came in CASP 13. CASP is a way in which you can put a competition. AlphaFold 2 came in CASP 14, which is like the next year competition, and it broke all the records. In that competition, 95% of the structures given to all the competitors were proven with full confidence. And this was a tool that came out of nowhere. In just few years, they just managed to ramp it, and now everybody is using it. There is like a lot of literature. Now I'm going to talk about it, that how this works beautifully. So for that, we need to understand what was the status before AlphaFold 2. So whatever you see as AF2 is the short form. So before AlphaFold 2, there were less than 10% of proteins which were available in the PDB database. Okay, less than 10%. So humans have roughly, think about it, if you're sitting in a classroom, I want you to guess how many proteins are there in human body. Okay, time is up. 25,000 around, and less than 10% would be so less. Okay, and that even then the whole proteins are not completely available. So what we do is that there are parts of the proteins that were available. So even then the 7% of residues which compensate the entire protein, only 17% had structural coordinates. That's called residue level coverage. Post AlphaFold 2 now, this has changed. Actually, I should put it almost 100% of the proteins are covered, which means that every species, every proteome of ant to plants to humans to mouse, all of them are there in AlphaFold 2 database. Okay, and the high confidence ones, residue coverage is 58%, which is a big deal. So what has got better? Now, if any biologist or any scientist who's working on a protein goes through a series of questions to understand that protein, let's say a maze. So what you see is a maze here, okay? There are folds that are not amenable to experiments. So meaning that there are certain proteins through which you cannot do experiments, but now because a part of it is available with high confidence, you can go ahead and do more hypothesis generations after you have the structure. You also have accuracy of structure higher, which I'll come to, and I'll convince you that there is like more number of structures now. There is a bad part of it as well, which I will speak, but let's say what has got better in this year. There are more starting points, meaning that now you have a structure, a protein with many kinds of complexes that are available in part and small and PDB, but now you have also their species level data. You have so much of data that you can now access. And again, this is wrong here. It's not 20 proteomes, it's actually 48 proteomes now, because recently there was additional paper which made it to full. Hello.
बोलिए.
There were at least 48 proteomes that were available. So now protein function in diverse cellular settings is available. Can somebody mute the person who's speaking?
If I've identified right, I have muted.
Okay, thank you so much.
Sorry.
All right, so now getting back to how does AlphaFold work? It's a bit complicated. So for school students, I'll try to make it as, but it's important for you to understand what is AI, how do people utilize the power of AI. So in this, if anyone is interested in reading the papers, these are the two papers, but let's start from the left-hand side. Every protein, I am sure you all would also know, that is composed of amino acids, okay? So this small differences, what you realize are different amino acids. And there is a database which the Google DeepMind company did. It had the genetic database search, meaning that particular sequence in fish, in rabbit, in chicken, and so on and so forth would make what we call as multiple sequence alignment. Okay. And then there is a pairing it did. Now this pairing was the most interesting input that AlphaFold 2 gave, meaning that it also computed that how these residues in all the species were connected. More technical word, this is called co-evolutionary matrix, that how certain residue pairs were coupled. Okay, this is very different, but although derived from MSA, but still very different form of pairing. Now in deep learning or in AI, if you are interested, you would know this, that what you give as an input or what kind of features you give and how you process that, what kind of tools you apply is so critical. So I think this was one of the fundamental things that AlphaFold 2 people gave. So prior to that, we were looking at MSA, that what were the previous homology, how is it connected to other species or other protein structures, which comes me to the last point, that it also had structure database, that what kind of previous structures are available that are close to this input sequence. So once this all is given, there is an MSA which is like represented, okay, and there is a pair representation. In that you have an complete Evo former, which is what they call as the AI module. And in this AI module, you go through, you have multiple blocks, and then you go as the last thing to give the structure. This is based on the coupling of the residues, on the previous MSA, on the previous templates. And then you put some amount of MD simulation as the last thing to refine the structure. And you do this 3 times to refine it again and again. So at the last, you get a 3-dimensional structure, which is modeled completely using the novel AI technology. Okay. And there have been huge, exciting developments since this has come. Scientists have realized that now they can detect the pocket better. You know what is a pocket? A protein has pocket to bind to drugs, to bind to ligands, to bind to small molecules, to bind to proteins and other proteins. And by this, you can modulate their function. So this becomes very, very interesting. That at the atomistic scale, they were able to predict the pockets very nicely. Okay. This came directly after 2021. There were structures of complexes that were happening very nice. So there's a whole story about this, that once they were able to do one protein, you can also do how it partners with its same protein or another protein. So this was coming as a very interesting post development kind of thing. And then the AlphaFold team also worked on it and gave AlphaFold 2 Multimer as a tool. And there were other things that they were doing called experimental model building. So, for example, the X-ray crystallography and the EM that I mentioned early on in my talk, they're the techniques that you study a protein experimentally, also need a model. And for that also, they have started to utilize the high confidence core AlphaFold 2 models. Okay, so this became really interesting. And also to everyone now, you can use AlphaFold 2 through a notebook called CollabFold. So you don't need to come to fancy research institutes. You don't need to have access to your own supercomputer. You can simply type on Google CollabFold AlphaFold 2 and you just need to paste, for example, hemoglobin protein sequence. It will give you the structure. There are other techniques also which are now relying on it, such as protein hallucination. So now you can design the protein. Earlier, people used to design one protein because it was taking so long to predict its structure using ab initio developments. But now you can have a series of proteins that you can design. And through this design, you can not be now restricted to few numbers, but you can now go to hundreds of peptides, hundreds of interactions. And that's what, folks here have done. There are other tools like Rosetta Fold and others that have come. Lastly, a very important point which I want to make is the following, that all of this has not led to all good structures, like any other technique. We need to understand that there are limitations, and AI especially is not different. It is also falling in some kind of lacuna. Now, what is that lacuna? People have bad structures because they give bad confidence scores. And then there is a category which they are in a funny way, in a silly way, calling it ugly, meaning that these structures have so little confidence that you cannot say anything about it. In fact, there was a huge development where people were even claiming that you don't need to do experiments and it's all now proven, but that's not true. There is a significant part of prediction of these proteins that cannot be interpreted because of the low confidence scores. Okay, let me give you an idea. The one confidence measure is PLDT. It tells you from 0 to 100 how confident the score is. The other is PAE score. Somehow that text is not coming out here, but yeah, PAE score tells you how a protein is aligned with each other. A dark green color represents good alignment, but look how the light green color alignment is here. So if a protein residue on the X and Y, which is around 1000 residue long, 1000 residues are there in this protein, you have sections of the protein which are aligning with each other in a domain to domain manner in a poor PAE score. Therefore, maybe you can say they are very flexible, so therefore there's a problem in prediction, but they are domains that are properly folded and you have very high confidence. Okay. So this had come in 2021. I'll just give you now a very interesting example of how we studied using AlphaFold. So we are studying a pathway called autophagy. Autophagy is a cellular pathway of self-degradation. And we do this computationally. Okay, so we study all the proteins. What you see here is that nodes are there as proteins. So we are interested in studying these proteins. And we said, oh, there are two students of mine, Shantanu and Nidhi. Nidhi, who basically drove this work, said, okay, let me just see how many structures are available. So what you see is that in gray, those structures were available for this pathway. So in any pathway, you have hundreds of proteins that contribute. So is here. And then you see in green color, they're unresolved protein, which means that they were not present in the PDB database that I have explained to you. They have not been determined using any experimental technique. So it becomes a bottleneck in biology. So she just mapped onto it the PLDT score. So she got all the structures from AlphaFold 2, which is what you can do using a simple search on the internet in the AF2 database. It's online. And once you download that, as I explained to you, there's a confidence score PLDT. If it is high, then above 70, then it's a high structure. And if it's a low, then it's low confidence structure. So we kind of said, okay, don't look at orange, look at only blue. And it was very interesting that blue resulted in 47% of additional coverage. What that means is that 50% was not available, but in that 50%, at least 47% was high confidence. This was massive for us, which meant that now you can ask new questions. Now you have the availability of the data and you can, study the pathways. I'm not going into detail. Now you can study initiation, elongation with much more clarity. And this work exploited other techniques, but what we did was we took these structures as starting structure. We ran molecular dynamic simulations, which is the next topic of my explanation, and then we did experiment. And believe me, this we did all so fast in less than 2 years, and usually we would take years and years to do so many structures to do MD simulation. But now we had the starting structures, right? So we had the access to it. So this made our study very, very interesting. And then at the end, whatever we proposed, we took to our collaborators who do experiments. They validated our so-called speculations or putative results, and we really got very interesting findings from it. So this was our own research applying AlphaFold 2, applying AI methodology and studying it. And but again, I'm saying MD simulation bits and pieces here, but what about it? What about dynamics? And I don't know how many of you have been to like large concerts where there is a conductor who's managing a lot of musicians who are playing different kind of instruments. And this is exactly what's happening in cell. Each and every person, I want you to imagine, is a protein here and has different kind of function. Some can do violin, some can do cello and other instruments, and you are all in school, so I'm sure you may have hobbies, or some do, some not. So, somebody likes flute. So these are all different proteins. They're all dynamically performing that. Nobody would sit, to do that, because then you don't have any function sitting in the choir. So you have a dynamic aspect to sitting, and also you are different. So there is some kind of interesting perspective here for you to understand that all the proteins move, but X-ray crystallography and electron microscopy enlarge, cannot capture the dynamics. There are experimental techniques that can do that, but they have limitations. So one of the ways, I'm not saying it's the most important way because I study, but I'm biased. So I am very fascinated by dynamics in general. So we do study it using computational, but prior to just introducing that, let me just show you some examples of dynamic, not just from the instruments, but actually real simulations. So this is when you see a membrane, you all have skin and you have different membranes, you have different lipids on it. So this is how they move. This is a protein called chaperone. Look how nicely, like a camera aperture, it's moving. It's like opening up. How interesting can that be? How coordinated can that be? This allows for the proteins to come inside the cavity. So this is a beautiful field of research where we try to see how proteins move. And some key points before you go ahead. I have, of course, say that biomolecules are always in action. So again, go back to the musician, kind of when you are sitting, you're always in action, maybe sometimes with less frequency, sometimes with high frequency. You also have processes that occur on different time scales. So there may be someone who's wanting to come with a guitar note on the first and just at the beginning. So there are different time scales at which these biological moieties operate. They also vary by sizes. Now, this is very, very important component. And at the beginning of my talk, I showed you how big some of the proteins and the time scales are. So they all move now inside the cell. So to do that, we have something called computer simulations. So the basic principle is the same as you would play in a game. You reproduce the behavior of the system in a manner that you create a room here. But what we do is that we create physiological conditions using a technique called molecular dynamics. It solves Newtonian equation of motion. So for each atom, it's like simple MA, which is the second law of Newton's. F is equal to MA. And F is connected to negative gradient of potential energy. And in many ways, potential energy can be calculated using how bonds are calculated, how angles are calculated, how different molecules are arranged in Coulombic interactions and van der Waals interactions. Overall, this gives you a trajectory. And that can give you an access to very interesting kinetics quantity. It can tell you about dynamics, how a protein is achieving the binding pocket in an accessible way to a drug, and so many other things. I'm not going into detail, but yeah, so MD simulations, the way the force field operates, there are a lot of interactions that we put into prior, which is called a force field. So you have a reference, and then for every protein based on this reference, you create structure, which is a starting structure, and then you let it run. For example, a non-bonded interactions is considered like van der Waals and so on. Again, I don't have time, but what I want to say is that MD simulations was acknowledged 10 years back, more than 10 years back, now 11 years back, by these three wonderful scientists. And I happen to be indirectly connected to one of them because my supervisor was studying with Karplus. So Martin Karplus, Michael Levitt, and Arya Varshal, they all had seminal paper in the field in the time when I was really very not born or very young, 1979. This was that time when they had studied 100 picosecond of protein, which is now for me like nothing. So we do a long simulation now. Moving ahead, what we do is that we give a biomolecule or a couple of biomolecules of interest to these supercomputers. This is one of the supercomputer image of IGIB data cluster. And then what we get out, as I just briefly explained, is an MD trajectory. Okay, and this MD trajectory then has potential to give us the questions that we can address. What kind of questions can we address? Well, we can address a lot of questions. We can address how a small molecule binds to enzyme. We can understand how a protein understands. We can also understand how protein sits on the membrane and with very massive assembly like virus and its interactions. Now there are very proof of concepts that we can also go there. So all this, which have very different interfaces, which have different properties, some of you who have now studied chemistry would understand Sometimes there's low affinity, sometimes there's high affinity. These are some potential things that you can study. I'm going to give you very quickly 4 examples, and just enjoy the simulations and trajectories. Don't worry about all the biology. For example, there's a protein called GPCR, and when you're happy, this is the protein that is responsible. Sometimes it binds to dopamine, and also you have other phases. When you're sad, it binds to a different ligand. So there is a inherently, for all your phenotypes, for all your action, there is chemistry inside our biology. Okay, so this all you can study. We publish this, and we try to explain how binding is modulated with different kind of ligands. And there is a shift in the protein shape. So now here at the 1 you see it like this, but here it moves there significantly, so that makes you sometimes accessible to other ligand sites. So the other protein which I want to say is that we are looking at very nicely is the process called autophagy and called LC3. How it sits on the membrane was of interest to us. What is the structural consequence? So what you see is a membrane. Okay, a membrane is composed of very different kind of molecules. POPC, like phospholipids, they're not there in the protein, they're there in the membrane. Look how interesting biology is. There are proteins that are composed of carbon, nitrogen, oxygen, hydrogen. The same molecules constitute also lipids, but the carbons, the way they organized, is a different molecule now. It's called a phospholipid, and how they interact make so much of critical functions in our body. So how LC3 molecules sit on the membrane was one of the questions that we address. I'll show you a small movie. So here, let's say this is the molecular simulation, sample simulation. I'm showing how it's sitting and how it's going inside the membrane. And once we understood this using simulation, we could actually do a lot of analysis, wrote our own programs to see how it's binding, and then we could find out what it is binding with. And then we went ahead and did experiments in collaboration with very, very interesting people who also like to do things together. So there is like then a choir that is formed, some folks who are interested in experimental validation with people who do simulations. We could give then the mechanism that this protein is sitting with cluster of specific residues and so on and so forth. The third example I want to give you is that how proteins interact with each other when they are sitting on different cells. Now there is a protein called Notch, which is binding to a ligand called delta or JAK, but there are 2 ligands. So how it binds determine how initially in the embryo different cells form. This is so interesting, and there is no information available because there is no structure. So we model the structure of Notch, and then this is the biology of Notch. When we model the structure of Notch, we explain we just set ourselves to understand how does it bind to the ligand. So there were many computational approaches we did. A little less time here, but you can just understand from this slide that how it was binding to its ligand was not just possible from one kind of simulations, but different kinds of simulations. It gives us different results. So see how interesting it gets. Like you can do different algorithms to study different questions. And when we do that, let's say I'll just run this movie. I hope you're able to see the protein Notch is in gray, but now it's the colorful protein is in the different colors, and it is the same. The red here and the red here is the same. The orange here and the orange here is the same, but the ligand it is interacting to is different. And I don't need to give you any pointers to show you that it's completely different. The way it is moving, the way its structure is, the way it is folded, the way it is moving across time, and the way its stability is all different. And their outcomes when it binds to this protein and this protein is different. So you can really understand mechanism of what is going on inside your body. And the last example, and the favorite example, which I have personally learned a lot during COVID time, is spike protein. As you know, the SARS-CoV-2 is decorated on outside by this protein called spike. This is the first interaction, you can say, that happens with the host cell, which is humans. And these spike proteins is what we modeled ourselves on the membrane, and we also were routinely monitoring the mutations of spike protein. You would now know this terminology, that there were variations that were coming out, delta and so on. So we were routinely doing that, and we were very, very intrigued to understand how these mutations on structures were playing a role. And we did MD simulations trying to understand which domain and can we now predict. And yeah, some of this work is published, so whoever is interested can look into it. But primarily it gave us an opportunity, or an impact of our work was very heavy here, the most heavy I have done in my throughout the career, because we were able to explain in our published work that why are we seeing what we are seeing. And that made people develop better antibodies, better understanding of why we are having the changes in the RBD domain, for example. With that, I'm just I think right on time and a little bit less interaction, but I hope you enjoyed the talk. This is my team. Recent picture of Holi for our team, not looking good, but yeah, we enjoyed. So we are a very, very fun, playful, joyful team. All what I showed was because of the lab members who you see, and some of them, like the work, Notch Protein Surbi is now a postdoc in UCSF. She's not here. And other alumni who have contributed to my initial part of the work. The funding is from CSIR, Wellcome Trust, DBT India Alliance, DST, and Intel. And thank you all.
Thank you so much, Lippy. That was really like a wonderful talk, in the sense it was very visually rich. We could actually see the simulations of so many proteins and how they act together in coordination. I really loved how far we have come in visualizing what happens inside cells. So students, if you can put in your questions, if you have any, on the YouTube chat box, or some of the students are also attending from the CSIR labs, as I've learned in the last 1 hour. So if they have questions, and if the CSIR scientists can enable them to speak, I mean, we would be delighted to take those questions. Okay. If not, then I will thank Lippy again for joining us today. And I am very, very confident that this talk, which is already up on YouTube and will be shared across social media again, will gather a lot of attention. Thank you very much, Lippy. And students, all those of you who have participated, you will get to see the quiz that is based on Lippy's talk now. Do participate in it, because that is going to be necessary if you want to consider doing the week-long project in May with us. So with that, I'll close the session today and see you again next Friday. Next Friday, we will be having Dr. Divya Sopati from CCMB talking about using AI to understand biology and prevent pandemics. And so let's come back again next Friday to learn what he does with these tools. Thank you, Lippy.
Thank you so much. Thank you, everyone. Bye-bye.